Assalamu alaikum. So in this lecture, it's going to be a long lecture. We'll be talking about transcription. Um, so let's start right here. So first, let's define what a gene is. There are many definitions for what a gene is. But what you will see here is actually my definition, sort of, just to simplify things. So a gene is basically the entire DNA sequence. Okay, so it's the entire DNA sequence that is necessary for the synthesis of a functional RNA. This RNA can be messenger RNA, ribosomal RNA, transfer RNA, long non-coding RNA, microRNA, all the different types of RNA molecules. Or, so, for the synthesis of a functional RNA or a polypeptide. Now, this polypeptide can become a protein, or it can be cleaved into smaller peptides, but functional peptides, okay? Like small, short hormones. So again, it's the entire DNA sequence that is necessary for the synthesis of a functional RNA. This RNA can be non-coding, meaning that it may not produce a protein, or it can be a coding RNA, which is basically messenger RNA. This gene can also be responsible for the synthesis of a polypeptide. This polypeptide can form a protein with a specific three-dimensional structure, or it can be cleaved into smaller peptides with function. Now, so basically, when I say the entire DNA sequence, this would include things like the coding region, which is exons, as well as other regulatory sequences like promoter, enhancer, and so on, or non-coding regions like introns, okay, as well as 5' untranslated region, 3' untranslated region, and so on. Another term for a gene is a cistron. And a cistron is basically, it's a genetic unit, okay, that can produce one polypeptide at the end from one messenger RNA, and we call this monocistronic mRNA. So basically, we're talking about a mature messenger RNA after processing, as we will see what will happen in eukaryotes. Or this cistron can produce different polypeptides, different proteins with different functions from one messenger RNA. And here I'm specifically talking about mature messenger RNA, and this is called polycistronic mRNA. So let's talk about the general mechanism of So transcription is the process of making RNA from DNA. Okay. Now, in DNA replication, we use both strands as templates to produce another DNA strand. Now, here for transcription, we use one of the strands for any particular gene. Okay. Now, both of them can be used in general for transcription, but for any particular gene, one of them is used. Okay, so in this case, the DNA would be a template. One strand would be a template. So basically, let's say here we have a region of DNA that has 3 genes. Okay, so we say that gene 1 is located here on this DNA or this position of chromosomes, loci or locus. This is the position of gene number 2, and this is the locus of gene number 3. Now, notice that for genes 1 and 3, the upper strand is used. But for gene 2, the lower strand is used. The other one is used. So what does determine which strand to be used? It's the promoter, the presence of a promoter. And a promoter is basically the binding site of RNA polymerase. That's what it is. Now notice something, that RNA, whether mRNA, long non-coding, ribosomal RNA, whatever, they are synthesized. All RNA molecules are synthesized from 5' to 3', and the template is read from the 3' to the 5'. So here we have the coding region right here, read 3' to 5' again, and this is the messenger RNA, for example, or RNA molecule, okay? And this is the strand that, this is the sense strand. This is the strand that looks just like the messenger RNA, except that in RNA we have U, and in DNA we have a T. Okay, of course we have the other differences. That is, in RNA, the substrates or the nucleotides are ribonucleotides, which is different than DNA, where the substrates and nucleotides are deoxynucleotides. Okay. So what enzyme do we need? We need enzymes known as RNA polymerases. And what they do is that they catalyze the formation of a phosphodiester bond between nucleotides, and the substrates for these enzymes are nucleoside triphosphates, ATP, CTP, UTP, and GTP. Okay, now the final product that is attached is basically a monophosphate, and the two phosphates are cleaved off the substrate, generating energy that is needed for the production of, or for the formation of, the phosphodiester bond. Now, there are differences and similarities between replication and transcription, so you can read these on your own. And there are differences and similarities between DNA polymerases and RNA polymerases, and you can read these as well on your own. So here's the thing. The RNA polymerase is actually a large enzyme, really large. Now it occupies large space, large region of DNA. As it synthesizes RNA, it moves again from 3' to 5', so the template is 3' to 5', that's one. Two, RNA is synthesized from 5' to the 3' end. As the RNA polymerase is synthesizing RNA, note that the two DNA strands are separated, that's one. Two, part of the messenger RNA, or the RNA in general, part of the RNA is hydrogen bonded to the template itself. So as the RNA polymerase is moving forward, you have separation of the older parts of the RNA and hydrogen bonding of the newer part of the RNA with the template. Now, here's a look at an actual gene that is going through transcription, two genes actually. And note that you have the RNA molecules extending outward. Okay, what you see here, this is the DNA. This thread right here is actually DNA. Now, so what you see in here is actually, so the question is, where is the starting point of transcription? Where is the beginning of the gene? And it's right here at this point. Because as the RNA polymerase is moving forward, you can see that the RNA molecules are getting longer and longer. So it ends right here, okay? And the other gene starts its transcription at this point, and it ends at the other end as well. Okay, now this structure, by the way, is known as polysomes. And this is an efficient way for synthesizing many RNA molecules from the same gene all at the same time. You can see this figure right here. There are so many genes that are undergoing synthesis. You can see this gene right here starting its transcription at this point. The end is at that point right here. You can see how the RNA molecules are extended outward and getting larger and larger. Same thing here. This is another gene right here. That's a third gene, begin, end, and so on. So let's go through now transcription in prokaryotes. And we usually like to start describing any molecular process in prokaryotes because it's simpler. So once we understand the base of any process, we can go into eukaryotic mechanisms then. So in prokaryotes, some genes are organized as operons, and these operons are polycistronic. Again, definition of a polycistronic mRNA or a polycistron is basically it's a genetic unit that produces one messenger RNA, and this one messenger RNA can produce multiple polypeptides, and eventually they would form proteins from different regions of the messenger RNA, and each one of them has a totally different function than the other one, but they collectively participate in the same mechanism. Okay, so an example is the lac operon. Another example is the trip operon. Trip stands for tryptophan. So the idea here is all genes needed for the metabolism of lactose are produced from the same operon. All genes that are necessary for the synthesis of tryptophan are produced from the same operon as well. So once a cell needs tryptophan, what it does is that it produces all the enzymes necessary for the synthesis of tryptophan all at once from the same messenger RNA, from the same genetic unit, which is the trip operon. Okay? So there's only one RNA polymerase in bacteria, and this RNA polymerase consists of multiple subunits, and these subunits are two alpha subunits. We have one beta prime, and we have an omega subunit as well. Now these are the subunits that are necessary for the catalytic activity of the enzyme. There's another subunit, and it's called sigma subunit. This sigma subunit is not necessary, is not required for the catalytic activity of the enzyme, for the synthesis of the messenger RNA, but it does have an important function. So remember how I said that a gene is synthesized, and the beginning of the gene and the strand that is used to synthesize this gene is determined by the promoter. So what is a promoter? A promoter is the binding site of the RNA polymerase. It is located upstream of the transcription start site. And usually the start site, the start site of transcription is indicated as a +1. So the first nucleotide of the messenger RNA is known as the +1 nucleotide, the +1 site. Okay? Now anything that comes right before it is known as an upstream region. Upstream. So as you're going forward this direction, you're going downstream. Okay? So this region that comes right before the initiation start site is known as the promoter. It is the region where the RNA polymerase sits on, and then it decides to start transcription starting from the +1 site. Specifically, the promoter contains two consensus sequences. What we mean by consensus sequences is that sequences that are present, the same sequence is present in different genes. So there's an agreement on this sequence. And this same sequence can also be present in different genes of different bacterial strains as well. Okay, so the consensus sequences are known as the -10 and -35. Why are they given these numbers? Because -10 is located almost 10 nucleotides upstream of the transcription start site. And this region is also known as the Tata box because it's rich with TATA nucleotides. The -35 region is 35 nucleotides away from the transcription start site. It is upstream of the -10 site and the +1 site as well. Okay, so these two regions are the important regions necessary for binding of the RNA polymerase to the promoter. So if there is mutation in any one of these nucleotides in between, nothing would happen. There will be no effect on RNA polymerase binding to the promoter and no effect on transcription. Now, so the sigma subunit. What does the sigma subunit do? Now, what it does basically is that the RNA polymerase finds the promoter with the help of the sigma subunit. So the sigma subunit, in other words, guides the RNA polymerase to the -10 and -35 sites. Okay? And once it finds it, it can be released, but not immediately. So what happens is, when we talk about the process of transcription or any other process in molecular biology, we like to divide it into different stages. So we can divide it into initiation, elongation, and termination. Now for initiation, what happens is you have the RNA polymerase associated with sigma. Sigma helps the RNA polymerase find the promoter. When it finds the promoter, it forms a complex known as the closed promoter complex. The RNA polymerase in bacteria opens up the two strands and it starts synthesizing RNA. As it moves forward a bit, as it synthesizes the first 10 nucleotides of RNA, the sigma subunit is released. That's it. Its role is done. Once it dissociates, it can go and associate with another RNA polymerase, guiding it to start synthesis of that gene or any other gene. It doesn't matter. Okay? So without a sigma subunit, the RNA polymerase can scan the DNA, but it cannot attach strongly to the promoter. It's the sigma subunit that helps the RNA polymerase attach strongly with high affinity to the promoter. So without it, the RNA polymerase can still function, but it would have a hard time associating itself to the promoter. So the efficiency of transcription goes down. Now, elongation continues afterwards. The RNA polymerase does three things. It unwinds the DNA, so it opens up the DNA as it moves forward. 2, it synthesizes the RNA. 3, it rewinds the DNA right behind it. So it does three things at the same time. So it's multitasking, multiple hands or arms. Now note, as the RNA polymerase is moving forward, you have the RNA, the newly synthesized RNA associating with the DNA template. And then it dissociates as it moves forward, as the RNA is getting longer. Then we get to termination. Termination is determined by a specific sequence in DNA. Sequence is known as the stem loop structure because it forms a stem and loop structure. Now notice that this stem loop structure is rich with G's and C's. So the stem is really strong. I mean, it has really high affinity. And it is followed by U, or A in the DNA template. So why is that important? What happens is, as the RNA polymerase is synthesizing RNA, you have the formation of a stem loop structure. And this stem loop structure destabilizes the interaction between RNA polymerase with mRNA and mRNA with DNA. What makes it worse is that the interaction between messenger RNA and DNA is mediated by A's and U's. And the interaction between A and U is weak because it's based on 2 hydrogen bonds, whereas note that the stem is made of G's and C's. So it is stable. Now, so the formation of the stem loop structure, also known as a hairpin loop, the formation of this destabilizes the interaction between these 3 components, the RNA polymerase, the messenger RNA, and the DNA, so they get dissociated from each other. And that's the end of transcription. So now let's talk about transcription in eukaryotes. So eukaryotic genes are a little more complex. We have a transcription start site, we have a promoter region, but we have regions within the gene known as introns and exons. Now, this is particularly true for messenger RNA, by the way. Okay, so it gets processed by removal of introns and the connection of exons to each other. Okay, we'll see now how it works. So, how about RNA polymerases? We have 3 RNA polymerases in human cells. These are known as RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 transcribes rRNA genes. RNA polymerase 2 transcribes protein encoding genes, that is messenger RNA as well as microRNA. And this is our focus, by the way. RNA polymerase 3 transcribes tRNA genes and one of the rRNA genes. So these RNA polymerases, by the way, are not like bacterial RNA polymerase. Now, the bacterial RNA polymerase functions as a helicase, right? So it opens up the 2 strands from each other, it unwinds DNA, it synthesizes RNA, and it rewinds DNA. So it's multitasking. This is not the case for eukaryotic RNA polymerases. They need help. And help is offered by proteins known as general transcription factors because they are not gene specific. They work on many genes. So they're known as TF, transcription factors. And then they are given a number like TF2, meaning that this transcription factor is specific for RNA polymerase 2. And then we have different proteins, and each one of them is designated by a letter, A, B, C, and so on. So we have TF2A is a general transcription factor for RNA polymerase protein A. Okay? Now they do the whole work. They do the work that we just talked about. So what they do basically is that they help guide the RNA polymerase to the promoter. So they function like what? They function like sigma subunit. They aid in pulling apart the 2 strands from each other to allow transcription to begin, like what? Like a helicase. And they push the RNA polymerase forward to begin transcription. So now they function like RNA polymerases. Well, how about the promoter region? Do we have a theta box and do we have a -10 and -35 regions? Well, we have something similar but still more complex. So yes, we do have a data box, okay? And we do have other types of promoter regions. Some of them are upstream, like the TF2B recognition element. And we have a promoter region that is located downstream, like the downstream promoter element. Note that this promoter is located inside the transcription start site, or inside the cistron itself, the gene. And we have another type of a promoter known as the initiator, and it includes the +1 transcription start site. Now, not all of these promoters exist for all genes. Genes can have 1 or maybe 4 of these promoter regions. So genes have different promoters. Again, things are more complex. Now what happens is you have the formation of what is known as a pre-initiation complex. This complex includes the RNA polymerase as well as the other general transcription factors like transcription factor 2D, 2H, and so on. Okay? So there is something else other than the core promoter. So all of these sequences that we talked about are known as core promoters, okay? That include in bacteria the -10 and -35. That's the core promoter. The Tata box, the BRE, and DPE, these are all known as core promoters. Now, genes have something known as promoter-proximal elements. So whenever you hear the word element, you know we're talking about a DNA or RNA sequence. Now, these regions are located upstream of the core promoter, and they are important for strong expression. So if we don't have them, can we have expression? Yes, but it's not really that efficient. We need this sequence. Now, it's not really important by itself. I mean, it's important because it allows, or it's binding site of other regulatory proteins that are gene specific, so they function for specific genes, and they increase the efficiency of transcription. Okay? Now, so it's almost like the promoter-proximal element is almost like an alternative to operons. Remember how we said you have in bacteria, you have operons, and you have the production of different proteins with different functions, but they participate in the same process, that is metabolism of lactose, synthesis of tryptophan, and so on. Well, what happens here is that you have the genes responsible for metabolism of lactose, for example, in eukaryotic genomes, located on different chromosomes and different places. And their expression can be harmonized and it can be unified by having the same promoter-proximal elements. So once we have a promoter-proximal elements for all of these proteins, you have a single regulatory protein that can bind to the promoter-proximal element of gene 1 and gene 2 and gene 3, inducing their expression all at the same time. So it's almost like an alternative to operons. Okay, so this illustrates exactly what I'm talking about. So an operon is the cistronic unit, genetic unit that produces multiple proteins from different regions of the messenger RNA, but they all participate in the same process, such as synthesis of tryptophan. But in eukaryotic systems, the same genes here can be located on different chromosomes, but their expression can be controlled by the same promoter-proximal element. Okay? Now, So you have something, so we do have general transcription factors, but we also have tissue-specific and gene-specific transcription factors. So here we're talking about the same gene. Now, since all cells have the same genome, okay, but they do not express all genes or the same genes as each other. So the same gene exists in skin cells, liver cells, and brain cells. But in skin, you have some expression, but not a whole lot. Why? Because you have the pre-initiation complex with the RNA polymerase and the transcription factor, general transcription factors. So you have some expression. But in liver, you have a lot of expression going on. Expression is really strong. Why? Because in addition to the pre-initiation complex, you have an activator that binds to the promoter-proximal element. Now, in brain cells, there's no expression for this gene. Even though you may have the pre-initiation complex associated with DNA, you have a repressor that binds to a region, a regulatory region upstream of the core promoter, suppressing expression. So you have tissue-specific expression of genes as well. And this is determined by promoter-proximal elements as well as regulatory proteins such as activators, repressors, and so on. Now, in addition to that, we have other regions, regulatory regions, known as enhancers. Now, these enhancers, again, they are specialized. They're binding sites for proteins that are specialized, gene-specific, cell-specific, tissue-specific, regulatory proteins. Now the enhancers, by the way, they can be located anywhere in the genome. Well, not really far away from the gene, but they can be located at a long distance from a gene. Not only that, we can change their position. We can change the position of an enhancer from upstream to downstream, for example. We can flip it, we can invert it, and it can still be functional. Why? Because there is a feature of DNA known as DNA looping, meaning that DNA can loop around. So whether you put the enhancer here or there, the DNA can loop and it would allow for expression to take place. So how does it function? Well, you have regulatory proteins that bind to enhancers, and these proteins can interact with the regulatory proteins that are present on the promoter, but interaction is not direct, rather it's mediated by a protein known as a mediator. So here we have a mediator protein. It mediates interaction between the proteins bound to the enhancer with proteins that are bound on the promoter or promoter-proximal elements. So these are okay. So right here, that's a hypothetical regulatory sequence of a mammalian gene. You can have, that's the gene transcription start site. You have the core promoter right here. You also have a promoter-proximal element, and you have enhancers. And these enhancers can be located anywhere around the gene. They do not have to be close to that gene. Now, in addition to enhancers, we have silencers as well. Okay, so silencers would interact with specific proteins, and these proteins would prevent the RNA polymerase and the pre-initiation complex from initiating transcription. So it's the opposite as enhancers. So how does initiation, how is transcription initiated? Well, what happens is you have the first protein that binds to the promoter is known as TF2D. This protein brings all of the other proteins. It recruits all of the other proteins to the promoter region. So it binds to the tata box, and it allows for all of the other proteins to bind to the promoter region one after the other. Now there's another protein that is important, and it is transcription factor 2H. This protein functions as a helicase. Okay, so what it does is that once it binds to the promoter region, it opens up DNA, allowing for the RNA polymerase to start transcription. Elongation then goes on, and this is, by the way, elongation is catalyzed by phosphorylation of the tail of the RNA polymerase. So the RNA polymerase keeps sitting on the promoter waiting for its tail to be phosphorylated. Once it's phosphorylated, it can then start moving and it can then start RNA synthesis. And this phosphorylation is catalyzed by transcription factor 2H, the same protein that functions as a helicase. It can also function as a kinase as well. So this is a multifaceted protein. It has multifunctions. Now, so synthesis goes on until we get to the termination sequence. The termination sequence in human genome for human cells is basically a sequence of AATAAA followed by, this is known as the poly-A signal, followed by a CA, followed by a GT-rich sequence. Okay, so once you have synthesis, so that's in the genomic DNA. Of course, it's the complementary strand that is synthesized. Okay, so once you have the messenger RNA having the sequence AAU, AAA, followed by CA, followed by GU, rich sequence, you have proteins that recognize the sequence. So what they do is that they cut between the C and the A, and this is followed by the addition of a poly-A tail. So the sequence for termination again is AATAAA, followed by CA, followed by a GT-rich sequence. Now, so eukaryotic genes are monocistronic because eventually you have one messenger RNA produced, and this one messenger RNA would produce a single polypeptide. Now remember that the single polypeptide doesn't necessarily fold into a single protein with a specific three-dimensional structure. Rather, it can form a protein, but it can also form shorter peptides that do have a function. Now, it just happens that genes in eukaryotic genes or human genes are large compared to the messenger RNA. And the reason is that messenger RNA goes on, it undergoes processing. And when the mRNA is first produced, it's produced as pre-mRNA that contains regions known as exons and introns. And exons are the protein coding sequences. So introns are removed and exons are connected to each other, and only exons are used for production of protein. Now, this process, the process of removing introns, is known as RNA splicing. So again, you have that's a gene that is transcribed. You have the synthesis of a pre-mRNA or primary transcript containing exons and introns, and then you have the process of splicing, connecting exons to each other and removing introns. And now you have a mature transcript. Now this mature transcript again produces one polypeptide only. Now the thing is with splicing is that you also have something known as alternative splicing. Now we're going to talk about it in the cell biology lectures in actin microfilaments. I'll give you an example of a tropomyosin. Now tropomyosin is a large gene. It's made of a lot of multiple exons. Now what happens is that not all exons are used for the production of tropomyosin. It is cell dependent. Okay, so for example, for smooth muscle cells, they take exon 1, 2, then 4, 5, 6. They do not take 7, They take 8, 9, 10. They do not take 11, 12, and 13. These are all the only exons that these cells need. Now, striated muscle cells have another opinion. They say, I want exon 1. They all have to have exon 1, by the way. We don't want exon 2, but we need exon 3. So it's different than smooth muscle cells, and so on. So from the same messenger RNA or pre-mRNA, you can have multiple mature mRNA molecules produced. And these mRNA molecules that are produced can produce different proteins. Now, all of these proteins are collectively known as tropomyosins, but they are tropomyosin isoforms. Okay, they all have the same function. They bind to actin, they regulate actin, but they do so differently. Okay, so tropomyosin would have slightly different functions in different cells, and tropomyosin isoforms would be regulated differently as well in different cells depending on their needs. Okay, now this statement right here, basically what it says is that you never have, for example, during processing, exon 4 coming before exon 3. They follow the same order. Okay, it's either that an exon exists or not, but never an exon is located or placed before another one or after another one. So you don't have exons 1, 2, then you have 6, then 4, 5, 7. You don't have that. Okay, so that's what it says. Now, so note that once we have a messenger RNA synthesized, or even during synthesis of messenger RNA, the messenger RNA undergoes processing. And there are three things that happen to messenger RNA molecules. And these are known as capping, splicing, and polyadenylation. We already talked about splicing. Now, the molecules that are necessary for the processing of messenger RNA are all bound to the phosphorylated tail of the RNA polymerase. So the phosphorylation of the RNA polymerase does not only promote the RNA polymerase to start and initiate transcription. What it does also is that it's the binding site of the processing enzymes and proteins, that as the RNA is synthesized, you have the proteins jumping on the RNA, modifying it. Okay? The first processing that takes place is capping. And capping basically is addition of a GTP in a reverse orientation. Okay, so it's flipped right here. It's added to the 5' end of messenger RNA. Okay, now why are mRNA molecules capped? Because capping is important for their stability. That's one. Capping is important to tell cells this is mRNA and this is the beginning of the mRNA molecule. This is the 5' end of the molecule. Okay, so it's almost like it's wearing a hat so that no one can degrade the RNA, the messenger RNA, from the 5' end. No one can hit it on its head. It's protected. And at the same time, by looking at the hat, everyone knows that this is a messenger RNA. The other thing is that capping is important because it brings on the other proteins necessary for splicing and polyadenylation. It's also important because it binds to a protein known as a cap binding protein, and it allows for the transport of messenger RNA out of the nucleus, okay, into the cytoplasm. So any mRNA is not transported out of the nucleus until it gets fully processed, including capping. And once you have a messenger RNA outside and it is capped, capping helps in translating the messenger RNA into proteins. Then we have, so we talked about splicing, and then we have polyadenylation. And polyadenylation is associated with termination of transcription. So remember how in you have synthesis of the sequence AAU, AAA in DNA followed by C in RNA followed by CA followed by GU-rich sequence. You have the polyadenylation proteins that recognize these sequences, and what they do is that they make a cut, and that would terminate transcription. And this is followed by the addition of a poly-A tail by an enzyme known as poly-A polymerase. And you have 200 A's added to the 3' end. Now remember that this poly-A polymerase does not need DNA as a template. So all of these A's are added without being part of the DNA or part of the gene itself. Okay, so no template is needed here. So why is polyadenylation important? Because it helps transporting messenger RNA from the nucleus to the cytosol. Remember how I said that mRNA is not transported out of the nucleus until it's fully processed? Well, that includes polyadenylation. Polyadenylation is important for translation. We'll talk about it in the translation lecture. And polyadenylation stabilizes messenger RNA. So whenever messenger RNA is degraded, it starts with removal of the poly-A tail. Then messenger RNA is transported out of the nucleus once it's fully processed. So whenever you have a messenger RNA that is defective, like interrupted RNA, RNA that is not spliced properly, RNA that is large as a result of a frame shift mutation, it cannot be transported out of the nucleus. So stability or mRNA does not last forever. So in bacteria it lasts for about 3 minutes as a half-life. Half-life of eukaryotic messenger RNA is about 10 hours, an average of 30 minutes, and they get degraded. And the first step in mRNA degradation is removal of the poly-A tail. Now the messenger RNA can become unstable. Once you have the removal of the poly-A tail, you have exonucleases functioning at the 3' end, eating the messenger RNA, or you have enzymes known as decapping enzymes. They remove the cap, and the exonuclease would start from the other end, from the 5' end. So let's talk about a phenomenon in eukaryotic cells in here. Okay. And this phenomenon is related to gene amplification. So what we see in here is that, let's say that we have 2 genes in here. What happens is that this part of the genome, this part of the chromosome, this part of DNA would get amplified. So you would have multiple copies of the same gene. And the chromosome would look puffy. It's like that's a puffy coat. Okay, it looks puffy in here. That's when you have amplification of DNA. So why is it that that cells do this? Why do they amplify DNA? Well, if they are put under stress, okay, for example, if you have cancer cells can escape resistance from methotrexate, okay, which targets the enzyme dihydrofolate, what cancer cells do is that they amplify dihydrofolate reductase so that you have a lot of products produced, a lot of protein produced from the same gene, because now you have multiple copies that methotrexate cannot inhibit them all. So cancer cells overcome methotrexate by producing more dihydrofolate reductase by amplifying the gene itself. The same thing happens in breast cancer cells. Okay, as in normal cells have limited number of receptor known as the human epidermal growth factor receptor 2 or HER2. Now, what cancer cells do is that they amplify the HER2 gene, producing a lot of HER2. Okay, and what happens is that now these cells become very sensitive to even minute, small amounts of the growth factor HER2, and this growth factor stimulates cells to grow and proliferate. So as a if a woman comes with breast cancer, and we check this breast cancer and we see that there is amplification of the HER2 gene, these women are given a drug that targets the HER2 gene, and this is a type of treatment of this cancer. So finally, let's talk about regulation of mRNA stability as a mode of regulation. And I'll give you an example of something known as the iron response or responsive elements, iron response elements. So basically what happens is that you have a certain sequence that is encoded into messenger RNA molecules for certain genes. Okay, or certain messenger RNA molecules. And these mRNA molecules are the ones that produce proteins that are responsible for regulating the levels of iron in our body, in our system, including ferritin, transferrin receptor, ferroportin, and divalent metal transporter 1. So these proteins are involved in regulating the level of iron in our system. Ferritin, for example, stores iron when iron level is abundant, and that's in liver cells. Transferrin receptor stimulates the entry of proteins into peripheral cells when these cells need them. Okay, so it's either that you will have high level of ferritin when you have high level of iron in the system, and peripheral cells would be then happy. Okay, in this case there should be low level of transferrin receptor, or if the level of iron is low, then peripheral cells need iron, they produce more transferrin receptor so that more iron would get into the cells, and at the same time the level of ferritin should go down because it's not the right time to store iron. Okay, so there should be a balance in the synthesis or production of these two proteins. It's either that ferritin is high or transferrin receptor is high. Okay, if one is high, then the other one should be low, and vice versa. That is, the opposite is true. And this happens, this regulation takes place by a protein known as the iron response element binding protein. Okay, and what this protein does is that it binds to this element and it regulates the stability of the messenger RNA or the rate of translation. If there's iron deficiency in peripheral cells, in this case, there should be production of transferrin receptor and no production of ferritin. Now, so what happens is that without iron being in the system, the iron response element would bind to the iron response element. The iron response element binding protein would bind to the iron response element that exists in the 3' region of the transferrin receptor messenger RNA, stabilizing the messenger RNA of transferrin receptor, producing a lot of transferrin receptor. Good? Now what it does in ferritin, it will also bind to the 5' end of the iron response element. So note the location of this element. So it exists in the 5' region of the ferritin messenger RNA, blocking translation. We're going to talk about this in the translation lecture. If there's a lot of iron in the system, then it's time to store this iron in ferritin. And it's time to reduce the level of transferrin receptor because peripheral cells don't need iron inside them. Okay, So what happens is that iron would bind to the iron response element binding protein, releasing it from the 3' end of the messenger RNA of transferrin receptor, destabilizing the transferrin receptor messenger RNA, reducing the level of mRNA, resulting in the production of less transferrin receptor. At the same time, iron binds to the iron response element protein, binding protein, releasing it from the 5' end of the messenger RNA of ferritin, allowing translation to start of the ferritin messenger RNA, producing a lot of ferritin, so that this iron can then be stored in liver cells. So you can see here, with one hit, with one stone we can hit two birds at the same time. So the same protein would stabilize one messenger RNA to produce a lot of protein from this messenger RNA, and it would inhibit translation from the other messenger RNA, reducing the level of protein of this that would be generated from this messenger RNA. At the same time, when there is a lot of iron, the protein is released, destabilizing one mRNA and inducing translation from the other messenger RNA, and this is how regulation works. So you can look at this slide. It provides a little more details about exactly what I just said. Allah ma'akum.